Hello students, welcome to Vedantu NEET English. This is Baswaratso, your biology master teacher. Good afternoon or good morning, each and every one of you. So students, today is going to be a quick session, rather a small session, where you do a very small topic called as your C2 cycle, C3 cycle and C4 cycle from the chapter Photosynthesis in Higher Plants. Now students, this video is from the famous request which all of you keep telling me, sir, can we have a quick revision of these three topics? And here we are. Okay, so with that being said, students, quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know in the chat, all of you. Quickly let me know if I'm audible and visible to all of you. Yes, quickly let me know, students. Now, before we start C2 cycle, C3 cycle and C4 cycle, we need to understand small basics about photosynthesis. Yes or no? See, can we start every high level complex topic without knowing a few basics ever? Obviously not. We cannot do that. We cannot do the injustice. So for the very same reason, can anyone tell me in the photosynthesis, does it happen in the case of plant cell or animal cell? In the chat, first of all. Very simple question. Now, what is the meaning of photosynthesis? The meaning of photo means light. The photo means light and synthesis means to produce. So plants are producing food using light energy. Using light energy, they're able to produce the food. And all of you know, all of you are aware that the photosynthesis, the process, the entire process takes place in the case of mainly in the case of plant cell. In the case of plant cell. Now tell me, students, in the case of plant cell, is the plants taking in oxygen or is it taking in CO2? Very simple question, right? Tell me, is it taking in oxygen or is it taking in your oxygen or CO2? You will tell me, sir, plants take in CO2. Okay, fine. Plants are taking in CO2. Now this CO2 right here is getting converted to, a form of carbohydrate. Can anyone tell me which is the simple carbohydrate which is found in the case of plants? You will tell me, sir, it is getting converted to glucose. Yes or no? It is getting converted into glucose. That is right. Now students, I will play a small game with all of you. A small game. That is, find the difference. Tell me, here we have carbon. Here we have oxygen. Here also we have oxygen. Can anyone tell me, are we adding something anywhere? Anyone, students, can you see? Is there any addition of something anywhere? Obviously, if you can see the clear difference, there is an addition of hydrogen. Yes or no? So can I tell from the difference, there is something called as addition of hydrogen. Addition of hydrogen, and addition of hydrogen, the process is called as what? The process is called as reduction. So students, the first point on the board right now about photosynthesis is that it is a reduction process. It is a reduction process in which CO2 is getting reduced to glucose. CO2 is getting reduced to glucose. That is the first step. Is that clear to all of you? Is that clear to all of you? Tell me. It is a reduction process. That is, the first point is that CO2 is getting reduced. CO2 is reduced. CO2 is reduced to glucose. Okay, beautiful. Now students, tell me, where is the hydrogen even coming from? See, I told you we are adding hydrogen here. Okay, but where is the hydrogen coming from? From air, randomly from the air? No. Then where is the hydrogen coming from? Now students, if you have a garden in your house, or if you have seen farmers working, you will always notice that they will water their plants. Yes or no? So can I tell this hydrogen right here is entering in the form of H2O, that is your water. And students, if you focus here, everyone, this particular hydrogen is given to your formation of glucose. If hydrogen is given to the formation of glucose, what is the fate of oxygen then? All of you know, plants take in CO2, plants release oxygen, and the same oxygen We breathe. Yes or no? We breathe the oxygen. All of you? Yes, we breathe the oxygen. Yes, we breathe the oxygen. So this oxygen is released out of the plant cell as a byproduct. As a byproduct. Does it make sense now, all of you? Does the photosynthesis make sense now to all of you? Plants take in CO2, they convert it to glucose. CO2 is reduced to glucose, and water is giving the hydrogen, and oxygen is released as a byproduct here. Now, apart from that, students, plants are making food, right? All of you tell me your favorite food. Tell me in the chat, veg food, not chicken, mutton and all. Tell me what is your favorite food. You'll tell me, sir, my favorite food is chapati, rice, curd rice, every single night. Crazy curd rice I will eat. Okay. So all of this food is organic in nature, which you are consuming. Now, students, plants are the magicians. Plants are the magicians which are converting inorganic food. They're converting inorganic substance into organic substances. Inorganic to organic substances. Now, at the same time, students, this entire process of your photosynthesis, does it require light energy? You're like, obviously, sir, without sunlight, how will they do photosynthesis? So, we will add a sun here. All of you remember sun. We will draw sun here. A nice for it. It's smiling. Okay. The sun is giving the sunlight. It is giving the sunlight. So can I tell the plants are absorbing the sunlight? Yes, they're absorbing the sunlight. Now tell me, students, if it is absorbing the sunlight, is it endothermic or exothermic? Quickly in the chat. You will tell me it is endothermic reaction. It is a endothermic reaction. Catabolic or anabolic? Anabolic reaction. Anabolic reaction. Why anabolic reaction? Because producing food. Beautiful. Now, students, in this entire reaction, is it enzyme involved? Yes, it's enzyme involved. Yes, it is completely enzyme mediated. It is completely enzyme mediated. So I'll give you a small recap what we just learned, all of you. Quick pay attention, all of you. CO2 is getting reduced to glucose, reduction process. Water is giving the hydrogen. Oxygen is coming as a byproduct. It is happening in the case of plant cell. It is an endothermic reaction, anabolic and enzyme mediated. From this slide, or this slide itself, many PYQs have come. Beautiful. This is how you understand photosynthesis, students. You don't by heart certain lines. You can't be like, sir, photosynthesis is a enzyme mediated process where we plants use sunlight in the form of chlorophyll and they produce organic substance. Nah. All that is not necessary. You learn this. This is more important. Okay. I believe all of you understood this. All of you understood the process of photosynthesis. The process of photosynthesis. Now, students, all of you know there's a famous equation, yes or no? That is 6CO2. So plants are taking in CO2. Plants also require hydrogen, yes or no? Plants require hydrogen, that is coming in the form of water. It is getting converted to what, students? Glucose, yes or no? It's getting converted to glucose in the presence of what? In the presence of sunlight, as well as in the presence of chlorophyll, and what are the byproducts? Tell me, students. The byproduct is again one of the water as well as your 6O2, which is released as a gas. This is your famous equation. So we call reduction process endothermic. No, endothermic because it is absorbing, and reduction process because addition of hydrogen is happening. Okay. Now, students, perfect till now. All of you understood till now, students. Now, before we go to C2, C3 again, let me tell you, you need to know where does this entire process takes place. Where is it happening? If I ask you, students, if I ask you the question, tell me, where is the entire process of photosynthesis occurring? You will tell me it is happening in the case of green plants. Yes or no? You will tell me it is taking place in the case of your green plants. Okay, green plants. Where exactly in the case of green plants? Tell me, students. Where exactly in the case of green plants? In the case of green plants, we have something called as leaves. In leaves, we have something called as mesophyll cells. Under mesophyll cell, students, under mesophyll cell, we have something called as chloroplast. Yes or no? We have the chloroplast. Okay. So, students, all of you know now, right now, all of you know, it is taking place in the case of chloroplast. Now, if you see the structure of chloroplast, tell me, students, is it a single membrane or double membrane? All of you know, similar to your Golgi complex, and as well as the nucleus, your chloroplast also is a double membrane structure. Yes or no? It is a double membrane structure. Okay. So can I tell we have something called as outer membrane and inner membrane? We have the outer membrane. As well as the inner membrane. Okay. Outer membrane as well as the inner membrane. Now, students, inside we have certain coin-like structures. Can anyone tell me what are these coin-like structures? These coin-like structures are called as thylakoid. They are called as what? Thylakoid. Okay, thylakoid. Now, these coin-like structures are not present as single. They are present in a stack. This entire stack is called as grana. It's called as grana. Okay, it is called as grana. Now, students, apart from that, this entire stack of coins, they are not present as single. There are many sets are there. Very rich it is. Very rich. So, let me draw more stacks. So, one is here, okay, and one is here. Randomly they are arranged. Okay, there are multiple grana. Now, you need to understand, students, all these grana are also interconnected. They're also interconnected. Now, this bridge or the interconnection is called as your stroma lamellae. They're called as what? Stromal lamellae. Or stromal lemma. Okay. Now, here we have a matrix. Also have a matrix here. The matrix is called as, can anyone tell me? We call it stroma. Now, why did I tell you Why did I tell you the entire story? Answer is very simple. The reason I told you this entire story is for you to understand the entire process of photosynthesis, right, is mainly divided into two categories. Something called as light reaction and dark reaction. And the C2 cycle, C3 cycle and C4 cycle are a part of your dark reaction. Then, and there is one more set called as light reaction. Now, where is it occurring and where is it taking place? Now, students, if you focus on this grana and stroma lamellae, as well as the grana, here we have the light reaction taking place. Light reaction. Here also we have the thylakoids, we have the light reaction. Then where is the C2 cycle, C3 cycle, everything happening? All that is happening in the case of stroma. Here we have the dark reactions. And this is the dark reaction which we are interested in today's class. This is gonna be the aim of today's class. And I believe till now all of you are able to understand. Grana or granum. Okay, granum we can call it usually. Okay. Now, students, this is your mesophyll cell. Can you see? Mesophyll cell. This is your dicot leaf because of palisade and spongy parenchyma. Chloroplast. Here we have your grana, and then we have matrix is called as stroma, thylakoid, and this is the entire structure. Now, the entire reaction is divided into two parts. Light reaction I will not be teaching you in today's class. We'll be focusing on dark reaction, which is also called as light independent reaction. I'll tell you one more thing, all of you. Listen to me very carefully. Okay. Just because it is called as a dark reaction, it doesn't mean it happens in darkness. Nothing like that. Just because it is called as a dark reaction, it doesn't mean it is happening in the darkness. Nothing like that. The reason it is called as a dark reaction because it does not require light. That is why it is called as dark reaction. Okay, dark reaction. But let me tell you, it is also called as light independent reaction. So please remember that. Because in examination, sometimes they will not give you as dark. They'll tell light independent reaction. Here we have light dependent reaction. Okay, light dependent reaction. Now see this, all of you listen to me carefully. In light dependent reaction, there is formation of ATP, there is formation of NADH. This ATP and NADH is used in your dark reaction. So ATP and NADH are used in dark reaction. Used in dark reaction to do what? To produce the sugar. Which sugar? The glucose. So in the entire reaction, students, in this reaction we learned here, right? This glucose formation is happening in the case of dark reaction mainly. In the case of dark reaction it is happening. Okay, so we have light reaction. I will not teach you. This is called as your light reaction on the board. Non-cyclic photophosphorylation. Here we have cyclic photophosphorylation. I will not be teaching you this today. Okay, the only thing you need to remember here is in light reaction we have NADH formation, ATP formation, and these two are used in your dark reaction. Now students, with that, can we start today's main topic, the main final show, that is your dark reaction now, also called as your C3 cycle, also called as PCR cycle, also called as Kelvin cycle. Can we start now? Can we start? Show some energy in the chat, all of you. Yes, all of you. Can we start now? That is called as your dark reaction now. Can we start? Yes, yes. Okay, amazing. Now listen to me very carefully now. It is also called as biosynthetic phase. Why is it called as biosynthetic phase? Because there is formation of glucose here, I told you just now. So biosynthesis of glucose. Also called as Kelvin cycle because of the scientist who was involved, Melvin Kelvin. Yes, does not require light. It requires products of light reaction, that is NADPH and ATP. Now tell me students, where this entire process taking place? Tell me students, where is it taking place? It is taking place in the case of stroma. All of you should know the basics. Occurring in the case of stroma. Okay, occurring in the stroma. It is also called as what? PCR cycle, photosynthetic carbon reduction cycle. Okay, carbon from the atmosphere is fixed to a sugar molecule now. Sugar molecule. So now we will start with your dark reaction. Raw materials: ATP, NADH, CO2. ATP and NADPH is produced from your light reaction. CO2 is coming from the atmosphere. Okay, there are three main steps: carboxylation, reduction, and regeneration. Three main steps in your Kelvin cycle. Can we start? Kelvin cycle, also called as your PCR cycle. Not polymerase chain reaction, photosynthetic carbon reduction cycle. Okay, photosynthetic carbon reduction cycle, also called as your dark reaction. Now students, from this point onwards, I want everyone to write down. From now onwards because it's very important. Okay, clear? Let's start. Beautiful. So all of you write down this entire thing. You need to write down. Okay, which color should I use? Let's choose white. White is always easy to read, right? Now students, the first thing we need to understand here is the CO2 is entering from the sky. Okay, CO2 is entering the jet. Okay, now CO2 is taken up by a substance called as, molecule called as RUBP. That is ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate. Okay, bisphosphate. Ribulose 1,5 bisphosphate is right here. Okay, ribulose 1,5 bisphosphate is right here. Okay, now students, understand very carefully. Understand. Okay, now RUBP is getting converted to one more substance which is 3 carbon called as PGA. It is getting converted to PGA. Okay, PGA. Now students, this is technically 3 carbon, 3 carbon. This is how many carbon? CO2. CO2 is 1 carbon, 1 carbon. Okay, 1 carbon. Now students, if I'm getting 3 carbon here, if I'm getting 3 carbon here and I have 1 carbon here, how many carbon this is supposed to be? See, if you're a scientist, back in the day and you're doing this experiment, okay, and you find that the first stable product, which is a 3 carbon, here it is 1 carbon. So technically here we should have 2 carbon, no? See, 2 plus 1 should be 3. Yes, technically 2 plus 1 should be 3 here. That is what many scientists were thinking that time. And they were trying to find a 2 carbon compound here. They were trying to find a 2 carbon compound. See, this is 3 carbon, 1 carbon. So this is, if this is 2 carbon, 2 plus 1 would be 3. Okay, it makes sense. It makes sense. But after many extensive research, after many failed attempts, they got to know that this RUBP right here is actually 5 carbon. It is 5 carbon and 5 plus 1 is going to be 6. 5 plus 1 is going to be 6, that is into 2 times 2, 3 carbon. So 6 molecules of carbon here, 6 molecules of carbon here. Everything is balanced. Everything is balanced here. Makes sense now? Makes sense. Now this first step is called as carboxylation. The first step right here is called as carboxylation. It's called as carboxylation. Now in this first step of carboxylation, there is an enzyme involved. Without this enzyme, it doesn't make sense only. If this enzyme is absent, this reaction cannot go at all any further. The enzyme which I'm talking about is called as your Rubisco enzyme, the famous Rubisco enzyme, students, Rubisco enzyme. The enzyme involved here is Rubisco. Rubulose, bisphosphate, carboxylase and oxygenase. Rubisco enzyme is involved in this entire process. Without Rubisco, it is not possible. Okay. Now we have 2-PGA. Okay. Now this 2-PGA will get converted to triose phosphate. Will get converted to triose phosphate. Get converted to triose phosphate. Triose phosphate. Now this triose phosphate again is also 3 carbon. It is also 3 carbon. Okay. Now this triose phosphate will eventually get converted to glucose, as we know, which we use and eat and survive. The entire world is surviving on. Convert to glucose. Now this second process is called as your reduction. It is called as reduction process. It's called as reduction process, and in this process there is utilization of 2 ATP. There's utilization of 2 ATP. 2 ATP is getting converted to 2 ADP. At the same time 2 NADH is getting converted to 2 NADP plus. They are being used. This 2 ATP is mainly because of phosphorylation, and this 2 NADH is mainly because of reduction of carbon. Okay. So this is what is happening in the second step, triose phosphate. Now what is the next step? See, we need this RUBP. Without this RUBP, the entire reaction cannot keep on happening. So this triose phosphate will again get converted back to RUBP, and this step is called as regeneration. It is called as regeneration. And this regeneration step also requires 1 ATP. 1 ATP is getting converted to 1 ADP. 1 ATP is getting converted to 1 ADP, and this is your entire C3 cycle, also called as your Calvin cycle. Now you'll ask me, sir, why is it called as C3 cycle? Why is the reason it's called as C3 cycle? Very simple. Can you see here? All of you can see here? This one. The first stable product here is your 3 carbon. This is the first stable product, which is 3 carbon. which is 3 carbon, and that is why it's called as what? It's called as your C3 cycle. It's called as C3 cycle. Once again, students. Chalo. Delivery method. Okay. Now, students, tell me, any doubt till now? Any doubt till now? This is your C3 cycle. Easy, no? Carboxylation, reduction, regeneration. Carboxylation, reduction, regeneration. First, we have your RUBP. RUBP is getting carboxylation, that is your getting converted to your PGA. PGA is the first stable product. Then it is getting reduced to triose phosphate, which is getting converted to glucose, and again it is getting regenerated to your RUBP. Again, it's getting converted to RUBP. Okay. Clear, all of you? Clear? This is your entire C3 cycle. C3 cycle. Okay, entire C3 cycle. Now, students, tell me, in this entire C3 cycle, for one CO2, for converting one CO2, how many ATP is required? Tell me. See, this is for one CO2. This entire thing is for one CO2. For this entire one CO2, we require how many ATP? Tell me. 2 plus 1. Tell me, students. For one CO2, we require. For one CO2, we require 2 ATP. At the same time, we require 3 ATP, right? 2 ATP in the second step, once the last step, one more ATP. So 3 ATP. 3 ATP and 2 NADPH. NADPH. Okay, for one CO2. Now, in examination, there's a famous question. There's a famous question in examination. They ask you for one CO2, this is the fate. Now, what about for glucose? For glucose, what do you do for glucose now? For glucose, see, this is for one CO2. For one carbon, this is for one carbon technically. Now, for 6 carbon, you need to do what? 6 carbon, you need to do multiply into 6. Into 6. Tell me the answer. 18 ATP and 12 NADPH. 12 NADPH. This is the answer for one glucose. This is the answer for one glucose. For one glucose. Done. For one glucose. Okay. Now, these are examples of C3 plants, that is your wheat, soybean, oats. In all of these plants where the C3 cycle takes place. C3 cycle takes place. Done. C3 cycle takes place here. Okay. Now, students, I will tell you something called as photorespiration. Ready? Ready, students? Ready for photorespiration? Very simple concept. I will teach in a very most efficient manner the entire photorespiration. Okay. Now, C3 cycle is done. That's all for C3 cycle. Very, very simple, easy cycle to understand C3. Okay. Now, photorespiration is even more easy. It is even more easy, photorespiration. Can we start? Can we start? Yes. Now, I'll give you a simple idea. Okay. For example, my name is, what is my name? Okay, let's say my name is Rubisco. Let's say my name is Rubisco. Okay. Okay. Let's say my name is Rubisco. I am right here. Okay. Now, on this side and this side, I have two people. Here I have something called as your CO2. Here I have your oxygen. Okay. So this Rubisco, students, this Rubisco has two main options here. It is called as rubulose bisphosphate carboxylase oxygenase. Yes or no? It's called as, one second, let me this. Okay. Rubisco has two main things. Here he has one CO2. Here I have oxygen. Okay. In normal condition, when CO2 and oxygen, when they are equal, who will I choose, students? Tell me. When your CO2 and oxygen both are equal, in the atmosphere they're equal. When they are equal, students, when they are equal, I will always go for what? I will always go for CO2. Okay. Higher affinity for CO2. Equal CO2. Okay. Okay. Now, students, understand. As soon as CO2 is reducing, in the atmosphere, I will leave CO2. I'm like, I don't know who you are, and I will go with oxygen. Oxygen is nice now. He doesn't have any money, so I'll go with oxygen now. Okay. So when there is less CO2, Rubisco will go with oxygen. Rubisco will go with oxygen. Okay. Now, what if in the atmosphere? When there is more, oxygen? See, there are two options, three scenarios. When CO2 and oxygen both are equal, I will always go with CO2. But as soon as CO2 is decreasing, I will leave CO2 and I'll go with oxygen. Now, again, in the atmosphere, if there is very high CO2, again I will go with oxygen. Three scenarios, equal, CO2. CO2 reducing, I will run to oxygen. Oxygen is more, I will go again with oxygen. So depending upon the situation, right? I will change my preference. Yes or no? Same thing is done by your Rubisco also. When CO2 and oxygen, when they are equal in the atmosphere, will go with CO2. As soon as CO2 is decreasing, Rubisco will run to oxygen. It will run to oxygen. As soon as oxygen is increasing, again Rubisco will run with oxygen. So this Rubisco has some little mental. Okay, I'm not Rubisco anymore. Okay, he will always prefer and he will keep changing his mind. Okay, Rubisco will keep on changing his mind always. Okay, now that leads to something called as photorespiration. Now, what do I mean by photorespiration? In the case of photorespiration, what is happening? Your RUBP is there, right? RUBP, okay, instead of forming a bond with CO2, it is forming a bond with oxygen. Why? Because of the enzyme that is Rubisco. Because carboxylase, oxygenase, it will produce oxygenase, and as a result of this reaction, we will end up getting something called as your Pg plus PGA. What is PGA? Same thing here. We learned right here, right? Phosphoglyceric acid, 3 PGA, 3 phosphoglyceric acid, 3 phosphoglyceric acid will get here. Again we'll get PGA. Now what is this PG? Again now? This PG is nothing but phosphoglycolate. Phosphoglycolate. PGA is normal. Okay. And tell me, students, how many carbon is RUBP now? Tell me, students. RUBP is how many carbon? RUBP is 5 carbon. How many carbon here? 0 carbon here. So here the PG is how many carbon generally? PJ is 3 carbon and this is 2 carbon. So 2 plus 3 is 5 and 5 carbon. Everything is balancing out. And this is called as photorespiration, where your RUBP forms a bond with oxygen to make PG and PGA. And let me tell you, students, this is a complete waste body reaction to your plants. Now in your house, you will only eat, you will not study, you will not do anything. You will simply sit, eat, sleep, repeat. Who will tell you waste body? Tell me, who will tell you waste body? In your house they will call you waste body. No, if you are not doing any work. Same thing is happening with your photorespiration. Why is it called as waste reaction? It is called as waste reaction because there is no sugar formation. There is no sugar formation. There is no ATP formation. There is ATP is being used here. ATP is being used here. CO2 is being released because oxygen is binding. CO2 is being released. So this is a complete waste of time reaction to the plant, but plants do not have any option. As soon as oxygen is going up, as soon as CO2 is coming down, the poor plant has to undergo your C2 cycle, also called as your photorespiration. Now, why is it called as C2 cycle? Because the first stable product is a 2 carbon. That's why it's called as C2 cycle. That is why it's called as C2 cycle. Okay. Waste body reaction, the C2 cycle. Now, students, understand this. This PG will eventually get converted to PGA. Yes, this PG will eventually get converted to PGA. But do you know how? The small extra information I'll tell you today. See, you know your entire C3 cycle happens in the case of chloroplast, stroma. But this C2 cycle is a little special. Why? Because this C2 cycle happens in 3 different organelles. It will start with chloroplast. It will start with chloroplast. That is formation of PG. Then the intermediate is sent to your peroxisomes. Then it is sent to mitochondria. And that is when the reaction is completed. This is a photorespiration which happens in 3 different organelles. It takes place in the case of 3 different organelles. That is your photorespiration, complete waste of time. The bigger question is, see, occurs in C3 plants, O2 is consumed and CO2 is released. Rubisco oxygenates RUBP. Okay, when does it happen? It usually happens at high temperature, low CO2 to O2 ratio. Or I'll just write for you. When does it take place? It takes place when we have more oxygen in the atmosphere, or we have very less CO2 in the atmosphere, or the temperature is very high, or light is very high. All of this happens when? All of this happens in the case of your C3 plants. But does it happen in the case of C4? No, absolutely not. It does not happen in the case of C4. That is the beauty of it. Now I'll tell you why. Listen to me very carefully. Let's go back to this reaction. I'll tell you. Okay. Now, imagine now, students, all of you think. I'll tell you why this photorespiration or C2 cycle does not happen in the case of your C4 plant. Let me tell you. Listen to me very carefully. Now, this Rubisco is very free. Rubisco is roaming around, say, and it has the opportunity to capture CO2, opportunity to capture oxygen. Now, it is very distracted. Now, students, you are also very distracted in the house. For example, you are spending a lot of time on Instagram and Facebook. Okay. Now, how do you remove the distraction? How do you remove the simple distraction? Simple distraction is delete Instagram or put a lock on it. Lock on Instagram. Yes. Or if you are getting distracted by your siblings who are coming into your house, coming into your room and troubling you, what do? You lock them in one room. If a dog is troubling you, you lock yourself in one more room. So you remove the distraction, right, by locking it away. Same thing we are doing here by locking it away. So what do you do here? Understand, in the case of your C4 cycle, we are making a locker room. Okay. In this locker room, we have the CO2. In the same locker room, we have the Rubisco. Now tell me, students, is there any option for the Rubisco to form a bond with oxygen anytime? Obviously not, because they're already locked in a room. Can you see? They are locked in a room, and where there is only CO2, when there is only Rubisco, there is no option. They should always bind. The Rubisco enzyme will along with RUBP, it It will always bind with oxygen, and we will always get there will be no photorespiration. There will be no photorespiration because of this locking mechanism. And this is seen in the case of your C4 plants. This is seen in the case of your C4 plants. In C4 plants, there is a chamber. In C4 plants, there is a small locker. In that locker, we only have Rubisco and we only have CO2 there. There is no way oxygen can enter anywhere near it. Not possible at all. Okay, so did all of you understand the difference between why your C4 plants are your smart plants? Did all of you understand why we call C4 plant as smart plants? Because they are making a small chamber there. Simply destroy mobile, huh? Yeah. If you destroy mobile, gone. Mobile only gone. You will also get one chappal in the house. Destroy the mobile, it seems. Did all of you understand? Here, in the case of C4 plants, we are making a locker. In that locker, we only have CO2 and we only have Rubisco. Nothing else is present there. Nothing else is present in that locker chamber. Okay. Now, understand how does it do it. So, in the case of C4 plants, there is a different type of anatomy only. In case of C4 plants, the anatomy only is super different anatomy. See this. How many of you know what is a wreath? How many of you know what is a wreath? This is called as a wreath-like arrangement. It is called as a wreath-like arrangement. Wreath means this. This is called as a wreath. Christmas wreath. It's called as a wreath. So we can see a wreath-like arrangement here. Okay, we can see a wreath-like arrangement here. Okay, can you see here? Here. This right here is the bundle sheets. Bundle sheets are arranged in a wreath-like structure. Bundle sheets are arranged in a wreath-like structure. This is called as wreath-like structure of bundle sheets. On the inside, we have the vascular bundles. Vascular bundles. So what is happening here, students? Nothing is happening here. Main thing is vascular bundles are surrounded. Vascular bundles are surrounded by two rings. Are surrounded by two rings. Can you see? Inner ring, outer ring. So this is the inner ring, outer ring. Vascular bundles are surrounded by two rings. The inner ring is called as made up of bundle sheet. The inner ring is made up of bundle sheet. It's made up of bundle sheet. Inner ring is made up of bundle sheet. And this bundle sheet has large chloroplast. It has large chloroplast and it has some kind of impermeable substance. There is no interstellar space here. Interstellar space is completely absent here. And it is completely impermeable. Nothing can enter here. Now tell me, students, if oxygen cannot enter into all, if oxygen cannot enter inside the gel, there will be no photorespiration. There will be no photorespiration at all if oxygen cannot enter inside. This is called as a wreath-like arrangement where vascular bundles are surrounded by two rings. Inner ring is basically your bundle sheets, which are completely impermeable to any form of gas exchange. Any form of gas cannot enter here. They're compactly arranged. It's called as a wreath-like arrangement. Also called as your Kranz anatomy, which is found in the case of your C4 plants. Okay, so bundle sheets are arranged in a form of wreath-like around the vascular bundles. Bundle sheet cells are characterized by having large number of chloroplasts, thick walls, no interstellar spaces. C4 plants can tolerate high temperature, respond to high light, lack photorespiration, provide greater productivity of biomass. All of these 4 points are a PYQ. It's a PYQ. Okay. And all of you, I almost forgot to wish you Happy Children's Day, all of you. I wish yesterday only I wished you all of you Happy Children's Day. Yes. Happy Children's Day, all of you. Now, students, you need to understand this C4 cycle is also called as hatch and slack. It's also called as hatch and slack pathway. Why? Because it was the scientist who discovered it. Hatch and slack. Here we have hatch, here we have slack. Hatch and slack pathway. And this hatch and slack pathway, also called as your C4 cycle, this mainly happens in the case of tropical plants. Mainly happens in the case of tropical plants. Like your tropical plants such as your, can anyone name any tropical plants? Tropical plants such as your, what is that? Maize, sugarcane, sorghum, amaranthus, right? Maize, sugarcane, amaranthus, all of this is where the C4 cycle takes place. Okay, there is 4 main steps, and let's draw it right now. Let's draw it. Now again, students, I will draw it for you. I want everyone to draw with me and write with me for a better understanding. Can we start? Can we start, students? Yeah, all of your child only. All of your child. Can we start? Yes or no? Okay. Let me draw it for you, students. Okay, there are 2 types of cells here. There are 2 types of cells here. There are 2 types of cells here. Okay, now one is called as your mesophyll cell. This one is called as your mesophyll cell. Mesophyll cell. Okay, now one of the bottom is called as your bundle sheath cell. It's called as bundle sheath cell. Bundle sheath cell. Mesophyll cell as well as your bundle sheath cell. Done. 2 different types of cells are involved here. Mesophyll on the top, bundle sheath on the bottom. Now what is happening here? Again, CO2 is entering. Yes sir, CO2 is entering. CO2 is entering. Now this CO2 right now, see, in previously CO2 was taken up by your RUBP. Yes or no? CO2 was taken up by your RUBP previously, but right now CO2 is taken up by a separate type of receiving carbon. That is, your CO2 is picked up by something called as PEP. That is phosphoenol pyruvate. What is PEP? PEP is called as phosphoenol pyruvate. Phosphoenol pyruvate. Okay, phosphoenol pyruvate. Okay, so the CO2 plus phosphoenol pyruvate. Now tell me, students, this CO2 is 1 carbon. CO2 is said to be 1 carbon, that's all. Okay, CO2 is 1 carbon. Phosphoenol pyruvate is 3 carbon. 3 carbon. As a result, we end up getting a 4 carbon compound called as oxaloacetic acid, which is technically 4 carbon, and this is the first stable product, 4 carbon. That is why it's called as C4 cycle. It's called as C4 cycle. Okay, now students, this what is PEP, phosphoenol pyruvate. What is OAA now? What is OAA? OAA is nothing but oxaloacetic acid. It is called as oxaloacetic acid. Oxaloacetic acid is the OAA. Now, in this entire process, there is an enzyme involved. Without enzyme, it's not possible. The enzyme involved here is pepcase. Pepcase is the enzyme. The enzyme involved here is the pepcase enzyme. Pepcase enzyme is the involved here. Pepcase enzyme is involved in the CO2 plus phosphoenol pyruvate will give you OAA PEP. That is PEP is involved. Okay, done. Now students, understand very carefully, all of you understand. This is very crucial step. Now oxaloacetic acid is converted to something called as malic acid. Malic acid. and malic acid is transported into bundle sheet. Malic acid is transported into bundle sheet. Malic acid is transported into bundle sheet. Now here malic acid gets converted to OA again. OA gets again converted back to your pyruvic acid. Pyruvic acid. Your malic acid and oxaloacetic acid, both of them are how many carbon? They are 4 carbon. 4 carbon. Again, this also is 4 carbon. But pyruvic acid is 3 carbon. It is 3 carbon. So 4 carbon, 4 carbon, 3 carbon. So can I tell 1 carbon is missing from somewhere? It is coming out. Yes, it is coming out. As you can see here, 1 carbon is released out here. 1 CO2 is released. 1 CO2 is released now. Now, what is the fate of this 1 CO2, students? Who is picking up this 1 CO2? This 1 CO2 is now picked up by your RUBP again. This one is picked up by your RUBP. That is inside your bundle sheet. Inside the bundle sheet, there is a C3 cycle going on. C3 cycle is going on inside the bundle sheet. So this CO2 will enter the C3 cycle. Again, CO2 plus RUBP will get PGA, triose phosphate. Same 3 steps: carboxylation, reduction, regeneration. All 3 steps are happening here again. It's happening here again. So if I tell a statement, the statement is very simple. C3 cycle takes place in case of all plants. True or false? Doesn't matter if you're C4, doesn't matter if you're C4 plant, C3 cycle will still happen in the bundle sheet. Now, if I tell you C3 cycle is happening here, C3 cycle is taking place here. So do we need the Rubisco enzyme? Yes, sir, we need the Rubisco enzyme, and that is why Rubisco enzyme is present here. Rubisco enzyme is present here. Rubisco enzyme is absent here. Here we have peptidase enzyme. Here we have peptidase enzyme. Here we have Rubisco enzyme. Rubisco. Now this pyruvic acid again is sent up. It is sent up. Okay. Pyruvic acid is sent. Pyruvic acid. Now this pyruvic acid again gets converted back to. This pyruvic acid gets converted back to PEP. It gets converted back to PEP. PEP, and in that step again there is requirement of 2-ATP. There is a requirement of 2-ATP here. 2-ATP is getting converted to 2-ADP. ADP. That is your entire C4 cycle. That's all. That is all your C4 cycle. I'll repeat again. First we have CO2. The primary acceptor of CO2 is PEP here. But in the case of C4 cycle, in the case of your C3 cycle, it was RUBP. RUBP is the primary acceptor of CO2. But in the case of your C4 cycle, the primary acceptor is PEP 3 carbon. CO2 plus PEP will give you oxaloacetic acid, which is technically 4 carbon. Enzyme involved is peptidase enzyme. This gets converted to malic acid. Malic acid sent to your bundle sheet. Malic acid gets converted to oxaloacetate, pyruvic acid. 4 carbon, 3 carbon. 1 carbon is released. 1 carbon is released there. And that 1 carbon undergoes something called as your C3 cycle. Pyruvic acid is sent up, and this gets again converted back to PEP for the entire cycle to keep on happening, and where 2-ATP is used to form 2-ADP. And this is mesophyll cell, this is bundle sheet cell. Mesophyll cell, peptidase, bundle sheet cell, Rubisco enzyme. That's all is your C4 cycle. Nothing difficulty in this. There's not difficulty at all. It's very, very, very easy to understand the entire process of your C4 cycle. Okay, very understood. Friends, any doubt till now? Any doubt in your C4 cycle? All of you wrote down in your books, also called as Hatch and Slack pathway. All of you wrote down? Done? Now, if that is done, now we will do a comparison. We'll do a small comparison between your C4 and C3. A small comparison. Can we start? A small comparison. Yes? Now see this comparison, all of you. Cell type in which Calvin cycle takes place. C4, it is mesophyll cell because there is only mesophyll cell in your C3, not C4, C3. But in the case of C4, Calvin cycle is taking place in the case of your bundle sheet. Bundle sheet, right here we saw. C3 cycle, bundle sheet, bundle sheet. Cell type in which initial carboxylation reaction takes place, mesophyll cell, mesophyll cell. Both is mesophyll cell. Initial carboxylation happening in mesophyll cell. Mesophyll cell. There also we have only mesophyll cell. How many cell type does the leaf has? Here only one, here we have two cell types. Mesophyll, bundle sheet. Which is the primary CO2 acceptor? Here we have RUBP, here we have phosphoenol pyruvate. Okay. Number of carbons in the primary CO2 acceptor? 5 carbon. This is 3 carbon. PEP is 3 carbon right here. PEP is 3 carbon. Okay. Now, which is the primary CO2 fixation product? Is your PGA, phosphoglyceric acid, which is your 3 carbon. That is why it's called as C3 cycle. Oxaloacetic acid is a 4 carbon. So C4 cycle. C4 cycle. Done. Does the plant have rubisco? Yes, both of them rubisco. Does the plant have pepcase? No. In C3 we do not have pepcase. In C4 we have pepcase. Okay. Which cell in the plant have rubisco? Here we have mesophyll cell on the top. Only mesophyll cell. Here it's only in bundle sheet. Because in bundle sheet your C3 is happening. In bundle sheet C3 is happening. Rubisco is present in bundle sheet. Bundle sheet. CO2 fixation rate under highlight condition, medium, it will work properly. Whether photorespiration is present at low light, no, low light it is not present because light is required. Okay, it is not present at all. Whether photorespiration is present at high light, photorespiration will happen here. Photorespiration will not happen here at all. Whether photorespiration would present at low CO2, yes. As soon as CO2 goes down, photorespiration starts here. But here there is no photorespiration. Whether photorespiration will be present at high CO2, no. Very high CO2, there is nothing like that. Okay, nothing like that. Nothing like that. Temperature optimum 20 to 25 degrees Celsius, 30 to 40 degrees Celsius. Very high temperature. Example: wheat, rice, maize, sugarcane, sorghum are the examples. Examples is very important, which you need to remember at any cost. Okay. Biodiversity. Okay, we will do. We will do biodiversity. Don't worry, it will happen. Okay. So that is your C3 cycle and C4 cycle at one go. Okay. Now students, that is the end of theory. That is the end of theory. Now do you want to solve some questions? Let's do some questions to understand whatever we learned till now is actually helpful in our NEET preparation. Yes? Yes students? Can we start with some questions practice now? Quick questions. Okay. Now we will follow the same NEET format now. Same NEET format. But a small twist. Because it's difficult to calculate the marks, right? So we will calculate the marks based on the, if you get one right answer, 1 mark. No negative marking. Enjoy today. No negative marking today. Enjoy. For every right answer, you will get 1 mark. For every wrong answer, nothing. Very limited. Today Children's Day, no. Gift. Children's Day, no. So don't worry. So no negative marking. Okay. So for every right answer, 1 mark. The first question on the board, right here. Go. First question on the board. How many molecules of ATP and NADPH are required for every molecule of CO2 fixation during Calvin cycle? Go. This is right here is a NEET 2024 question this year right now on the board. Tell me students, how many molecules of ATP and how many molecules of NADPH we need in every Calvin cycle? Yeah, great gift. What can I give you, huh? Tell me. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Time's up, students. Time's up. Time's up. Time's up. Time's up. Tell me, what is the answer? What is the answer? See, we have 3 molecules of ATP. We have 3 molecules of ATP and 2 molecules of NADPH. So option number 3. Option number 3. So all the students who told the right answer, keep the track of your marks, and here we go. Okay. Next question on the board. Next question on the board. Okay. Next question on the board. In C4, in C3 plants, some O2 binds with rubisco, hence CO2 fixation decreases. Okay, yes or no? Yes or no? That in some C3 plants, CO2 will bind to rubisco because of what? Because of photorespiration. Because of photorespiration, it is happening. In C4 plants, mesophyll cells show very little photorespiration. Mesophyll cells show very little photorespiration. Is it true? Tell me, students. Does it show any little photorespiration? Does it show? In case of your C4 plants, is there photorespiration? Nothing. There's no photorespiration in your C4 plants. So what is the answer? Tell me now. What is the answer? See, this is correct. Correct. But is the second statement correct? No. Yes or no? Yes or no? Madhumiti, wrong answer. It's not 3, it's 2. Why? See, it's correct. That is, in C3 plants, O2 is binding with rubisco. Hence, CO2 fixation is decreased. In C3 plants, in the case of your C3 plants, what do we have? In the case of C3 plants, we have photorespiration. We have photorespiration is happening in C3. But it is not happening in the case of C4. Not happening in the case of C4. Okay. So 2 is the answer. Answer marks in the chat, all of you. Marks in the chat. Next question. Next question. Again, a very, very good question. I'll give you 20 seconds. Take your time and answer. Take your time and answer. Okay, take your time and answer. All of you. C4 pathway. Light reaction, photorespiration, and Calvin cycle. All of you remember studying phosphoglycolate anywhere? The PG. Remember PG? We studied about PG in somewhere, no? In photorespiration. So C is going to be what? C is going to be what, students? Tell me. C is going to be 4. C is going to be 4. Okay. A is going to be 3. Carbon molecule, phosphoenol pyruvate. We learned here. Remember? C4, PEP, phosphoenol pyruvate. So A is going to be what? A is going to be 3. A is going to be 3. Okay. Then you tell me. Light reaction is what? In light reaction, there is photolysis of water. In light reaction, there is photolysis of water. In your non-cyclic, okay. Kelvin cycle, RUBP. In Kelvin cycle, answer number 3. Done. Okay. Next question. How many molecules of, same question is repeating. Okay. How many molecules of ATP? How many molecules of ATP and NADPH are required respective of fixation of every CO2 molecule in Kelvin cycle? Again, easy question, students. 3 ATP and 2 NADPH. 3 ATP and 2 NADPH. Again, NEET 2024 question. Same question is repeating. Next question. Which of the following are required for dark reaction in photosynthesis? Which are required for dark reaction? Marks in the chat. For dark reaction, what is requirement for dark reaction? Tell me. Requirement for dark reaction. Do we need light? We don't need light. We need chlorophyll? No, no chlorophyll. Do we need CO2? Yes. We need ATP? Yes. NADPH. What is the answer? CDE. CDE. Yes or no? CDE. Yes, CDE. See, we need CO2 for fixation. We need ATP, NADH for energy. Right? Next question. PEP is a primary CO2 acceptor in phosphoenol pyruvate. Is it in the case of your C3 cycle or C4 cycle? CO4 plants or CO3 plants? Tell me, students. Phosphoenol pyruvate, we just now learned, students. phosphoenol pyruvate plus CO2 will give you OAA, oxaloacetic acid, in your C4 plants. In the case of your C4 plants. C4 plants. Next question again. RUBP, pepcase, cytochrome C. This is part of your electron transport chain. This is part of your glycolysis. So let's see if we can answer this question. This is a very good question. This is a mixture of two chapters. See, RUBP, you know, right? RUBP, you know, does it involve in reduction of water molecule? No. Catalyze the phosphorylation of glucose? No. Beta carboxylation of phosphoenol pyruvate in your C4 plants? In your C4 plants, does it do something? Does it do something in your C4 plants? Yes or no? Tell me. So here is the first one. So AS3 means only one option is there. Only one option is there, AS3. So AS4. Oh, capture atmospheric. Sorry, no. Beta carboxylation is different. It is pepcase. Sorry, pepcase. In C4 we have pepcase. Pepcase in C4. Yes. A is going to be 4. B is going to be 3. Okay. -1 for me. Okay. Next question you see. Student, this is again same question. How many ATP and NADP are required for synthesis of 1 molecule of glucose during Kelvin cycle? 1 molecule of glucose during Kelvin cycle. Go. 1 molecule of glucose. Do into 6 basically. Into 6. See, in normal condition we have, normal condition for 1 CO2 we have how much? 1 CO2 we have 3 ATP and 2 NADH, right? 2 NADH. Do into 6 into 6. You'll get the answer. Into 6 into 6 you'll get the answer. Tell me. Into 6 into 6 you'll get the answer. Done. This is a good question again. When read the question again, all of you, read the question. Very, very good question this is. Okay. When CO2 molecule is fixed as 1 molecule of triose phosphate. Triose phosphate is basically happening in the case of your second step, reduction step. Which of the following photochemically made high energy chemical intermediate are used in the reduction phase? Students, tell me. In reduction phase, what is used? How many ATP? And what is happening? Tell me. In reduction, second step, 2 ATP and 2 NADH. Second step. Done. The primary CO2 acceptor in C4 plant is phosphoenol pyruvate in C4, and it is found in mesophyll. Friends, tell me, is it found in mesophyll or bundle sheet? Mesophyll. Okay. Mesophyll cells have C4 plants lack Rubisco enzyme. Mesophyll cell on the top lack Rubisco enzyme. Yes, makes sense. Yes or no? Makes sense? Yes. Okay. So both are correct. Next question. The first stable product in your C2 fixation in sorghum. Students, sorghum. Remember sorghum. Sorghum is an example of your C4 plant. C4 plant. Then what's the answer? Tell me. What is the answer now? C4 plant should be a 4 carbon compound. 4 carbon compound is oxaloacetic acid. Yes, oxaloacetic acid. The oxygenase activity. Now your Rubisco is doing a double acting. Okay, double acting. The oxygenase activity of rubisco enzyme in photosynthesis, photorespiration leads to the formation of one molecule of 3 carbon. Do we get one molecule of 3 carbon? PGA. We get PGA, okay. One molecule of 6 carbon, no. One molecule of 4 carbon, one molecule of 2 carbon, no. Two molecules of 3 carbon, no. What do we get? In photorespiration, we get PG and we get PGA. 3 carbon, 2 carbon. 3 carbon and 2 carbon. This is 2 carbon, this is 3 carbon. Yes or no? Yes. Right? So answer is easy now. 1 is the answer. 1 is the answer. In Hatch and Slack pathway, that is C4 pathway, in C4 cycle, the primary CO2 acceptor is, who is accepting the starting? Primary CO2 acceptor is going to be, very easy question again, PEP. PEP, phosphoryl pyruvate. Phosphoryl pyruvate. Okay. And students, that is the end of today's session. Okay. So all of you understood the entire process of your C2, C3, C4 done and dusted. Now tell me in the chat all of your marks. What is your marks? Now students, I'm telling you again and again, the Children's Day offer is today. So go check it out. Go to the description of the link. There's a Children's Day offer going on. I've already made a video yesterday. So go check it out and you'll get a clear idea about what exactly we're doing at Vedantu, how we can help you in your preparation. So go check it out. The link in the description. And also let me know in the chat which is the next small packed session you want, where we do a small question practice and we solve some small concept and we teach you small packet of marks for all of you. Okay, so let me know in the comment section which is the next topic which you need. I was thinking we will do transcription, translation and replication. What do you say, students? The next small packet of topics should be replication, transcription and translation. Hopefully these 3 we can do in the coming week. If not, we can do some other things also. Okay, so with that being said, students, thank you so much for today's class. I will see you in the next video. Take care. Let me know in the comment section how was today's class. Do not forget to comment. Let me know in the comment section how was today's class. Thank you so much and bye bye all of you. Take care. Okay, bye bye.